Good morning again, everyone. We have a PhD thesis defense today. Julijana Cvjetinovic, supervisor Professor Dmitry Gorin and Professor Alexander Karsunsky. Let me introduce also jury members. We have online Professor Dusan Losic, University of Adelaide, Professor Vasily Fedotov, Skoltech, Professor Юлий Котелевцев, Skoltech, Dr. Andrei Sapelkin, Queen Mary University of London, Dr. Ilya Turchin, Institute of Applied Physics, okay, myself. So we have a usual agenda for PhD thesis, and we are starting with candidate's presentation. Julijana, 40 minutes plus minus. Yeah, not yet.
I did it. Okay. Can you hear me now?
Yes.
Great. Good morning, everyone. Thank you very much for the introduction, and thank you all for coming here today and also for joining us online. I am truly honored and a bit terrified to stand before you today as I present the culmination of 4 years of research and dedication, my PhD thesis on the topic of optical and mechanical properties of diatom structures. Before we dive into the specifics, here is the outline of my talk. So I will start with a short background to introduce the main objects of my research. Then I will explain the whole idea behind this thesis, thesis structure, methodology, the main goals. And then I will spend the majority of time by showing the main results that I obtained during these 4 years. At the end, I will make some main conclusions and also show the lists of publications, conferences I attended, and acknowledgements. I wanted to start this presentation by showing this mesmerizing video that unveils the hidden world that lies beneath this diatom cell wall. And as camera pans from the base upwards, you are greeted by this silica structure that defies simplicity. So at the very start, at the bottom, you will see the delicate lattice of pores that are arranged in a mosaic pattern. Then, nestled within this elegant framework, you will discover these vibrant green chloroplasts that are responsible for harnessing the energy of the sun and therefore responsible for the photosynthesis. But as you further ascend, you will encounter the nucleus, which is the epicenter of the genetic information and cellular regulation. Also, the journey doesn't end up here because you will discover these small lipid droplets that store the energy and they are ready to fuel the diatom's journey through life's MSET flows. So basically, this video is a testament for the breathtaking complexity that lies within seemingly simple diatom cell. And it reminds us that even in these tiniest beings on our planet, there is a world of complexity waiting to be discovered and appreciated. Diatoms are extremely diverse class of microorganisms with exoskeletons composed of highly porous silica called frustules. Their beauty is impressive, but their ecological significance is staggering. Not only they anchor marine food webs, but they also have an outsized atmospheric impact. They siphon out harmful gases like carbon dioxide out of the air and produce massive stores of oxygen as they photosynthesize. It is estimated that roughly 1/4 of the air we breathe is produced by diatoms. As they photosynthesize, they produce biomass rich in proteins, lipids, and other valuable molecules. And at the same time, they represent a source of a highly porous amorphous silica that can find various different applications in different areas of science. Their beauty fascinated even Charles Darwin, who mentioned them in his work on the origin of species. He asked, were they only created to be admired under the microscope? So his question tickled my mind, and throughout my PhD thesis, I tried to answer it. Diatoms attract generations of scientists of different specialties owing to their fascinating structure, abundance, functionality, and properties such as good biocompatibility, easy modification, thermal stability, high mechanical stability, and optical properties. And in here, you can see that diatoms are usually classified based on their shape into pennate, which have this bilateral symmetry, and centric diatoms, which have radial symmetry. And in this combination of interesting structural property, structure and interesting optical, mechanical, and other properties make them a suitable candidate for different applications in modern science and technology, including bio and nanotechnology, biomedical applications, environmental technologies, applications, as well as nutritional, industrial, and many others. Diatoms can be successfully reproduced in bioreactors and grown in lab conditions, giving the appropriate amount of light, temperature, nutrients, and salinity in the case of marine species. But in order to ensure the optimal biomass, it is crucial to understand how they grow and to monitor their growth phases. This, in the current time, the most conventional method implies the cell counting under the microscope. And this technique works well, but it is time consuming. It requires the extraction of diatom from the medium. Also, in the case of small diatom species, as shown here, after several days, it practically becomes impossible to calculate them due to the formation of cell clusters and so on. Therefore, there is a need to develop new methods to apply new approaches that could help us to monitor their growth. And the first part of this thesis actually explores the novel optical methods that could help us to monitor the growth of diverse diatoms, freshwater, pennate, and marine ones during long-term cultivation. As I have already mentioned, and you saw their remarkable structure, besides such structural diversity, they also possess intriguing optical and mechanical properties. When it comes to mechanical properties, there is a lot of literature, but still many questions remain unresolved. And that is why in the second part of this thesis, I decided to study mechanical properties to actually perform a comprehensive study that would involve cleaned frustule, which means without organic material, with the separate contribution from each layer. Then to also perform the study on wet and air-dried living cells, this time with all organic material trapped inside, from micro to nanoscale, and under static and dynamic loading conditions, which could be very useful for the future applications in various devices. But their optical properties are also very interesting considering such hierarchical periodic structure. And there is a lack of data, for example, if we are considering this frustule in their natural aquatic environments. Usually numerical simulations are performed in the air, not in the water. And we decided to study the impact of such periodic multi-layered structure on the distribution of light intensity inside this structure, and maybe to try to find a correlation with chloroplast distribution inside the diatom cell, which would further help us to better understand the photosynthesis processes. So this actually study that combines optical and mechanical properties is not only important for fundamental research, but also for future applications as functional devices in different devices, including MEMS ones. But I will talk about it later. Also, we can modify the surface of diatoms using different approaches. One conventional approach, and very interesting, for example, if we use nanoparticles. So in that way, we can modify the surface for enhanced properties, but still there is a need for improved control over the deposition processes of these for example, gold nanoparticles, silver nanoparticles, and others. We need to achieve uniform coverage, and also we need to be able to create a multi-layered and functional coatings. So the third part of the thesis actually explores a novel approach to modify the surface of diatomite powder with gold nanoparticles. That is why the main goals of these theses are monitoring of the growth of diatoms using optical and spectroscopic methods, study of optical and mechanical properties of individual cells, and functionalization of diatomite powder with gold nanoparticles. So basically, I conducted the research on 3 different levels, starting from colonies in suspension to individual living cells, as well as clean frustules, and then going to the diatomite powder. And that brings us actually to the thesis structure, which I divided into 9 chapters, starting from the introduction, literature review, experimental section, and then going to this main part, which includes monitoring and characterization of diatom colonies, visualization of individual diatoms, mechanical, optical properties, modification, and then finally at the end we have conclusions. You will see this schematic illustration a few more times, as it will indicate at what level we are for easier understanding. Basically, this slide illustrates the research design. So as I have already said, we have conducted research on 3 different levels. As for colonies in suspension, we monitored the growth using optical spectroscopic methods, as well as the standard cell counting. We also, for the purpose of monitoring the growth in bioreactors, developed a setup that we tested for monitoring changes in the concentration. We applied for the first time here photoacoustic imaging. All results were verified using spectroscopic techniques. Then when we moved to the study of individual diatoms, I applied different advanced imaging techniques to visualize them using, of course, scanning electron microscopy, transmission electron microscopy, fluorescence lifetime imaging microscopy, confocal microscopy. And then when it comes to the mechanical properties, we used atomic force microscopy indentation, in situ nanoindentation inside the column of scanning electron microscope as well. And we also conducted some numerical simulations of vibrational properties. If we are talking about optical properties, here we apply the Fourier model method to calculate the optical properties, and some data were obtained also using extinction spectroscopy and fluorescence lifetime imaging microscopy. In the last part, modification of gold-coated diatomite was performed using a combination of layer-by-layer assembly and freezing-induced loading. And we characterized the obtained hybrid samples using SCM, TEM, photoacoustic imaging. And at the end, we tested the capabilities of such system as a platform for surface-enhanced Raman spectroscopy. So basically, we believe that out of this first part, we can develop techniques that can be useful for monitoring the growth of diatoms, not only in the lab conditions, but also in their natural environment. Then when it comes to this study of mechanical and optical properties, the results obtained can help us in development of novel devices. It gives us valuable insights into the material designs, including maybe developments of MEMS microphones. And at the end, when we are talking about these composite materials, it would also be very useful for us to, in the future, develop the composite materials for example, sensing applications. Without further ado, let us embark on the journey to explore this intricate world that lies beneath the diatom cell wall. And I would like to start with the first part, monitoring and characterization of diatom colonies in suspension. First, for the purpose of cultivation of diatoms under control conditions, we fabricated a small bioreactor which consists of two main units. It consists of the control unit, which allows us to regulate the temperature, the light intensity, and the day-night regime. And also it consists of this growth chamber inside which flasks with diatoms are located. So first we started with the freshwater pennate diatoms, these small Acanthidium sibiricum cells, which have approximate size of around 10 microns. And we performed the cell counting during the first 4 days, where we observed steady growth of the culture. But after that, it became practically impossible to further monitor their growth due to the formation of cell clusters, which leads us actually to the exploration of novel optical methods for such purposes of monitoring of the growth. And here we applied the fluorescence visualization using IVIS spectrum CT in vivo imaging system and photoacoustic visualization with the help of raster scanning optoacoustic mesoscopy setup Explorer P50. What we see here is actually that the signal increases with increasing cultivation time up to 45 days. And then we observe this slight decrease. So basically the growth phases of diatoms include lag phase, exponential phase, stationary, and then dying phase. So by using this method, we could actually observe all these phases in the case of freshwater pennate diatoms, and the same results were obtained using the photoacoustic imaging. These results were verified using extinction and fluorescence spectroscopy methods. We also used the same techniques to monitor the growth of marine-centric diatoms. In this case, we have two different species. And what we see is that in this case, the maximum signal was obtained after 30 days of cultivation, which is slightly less than in the case of freshwater pennate diatoms. Also, fluorescence spectra shown here demonstrates the same results as obtained using fluorescence visualization with IVIS fluorescence system. Photoacoustic visualization in this case was very interesting. Why? Because we were able to actually obtain the signal from single cell, which was not the case in the previous diatoms. Why? Because the size of this cell is around 220 microns, and the axial and lateral resolution of optoacoustic microscopes are 10 and 40 microns respectively. So basically what we see here is actually one cell. So we did not obtain the signal from the whole colonies, but rather from a single cell. My very first published article was in photoacoustics, where we for the first time demonstrated the dependence of the photoacoustic signal on concentration of diatoms after performing a serial two-fold dilution. What is interesting here, besides the fact that it was conducted for the first time, is the fact that we showed that, for example, the minimal signal can be obtained when we have concentration of particles 3 cells per microliter. Those cells were also very small. Their size is less than 10 microns. So it basically shows us that we were able to obtain the signal around when we have like the actually at the resolution point of 40 microns. But also I would like to pay your attention to this solution of a photoacoustic wave equation, which shows us how the illumination pulse associated with the optical fluence, associated with the illumination pulse gets converted into a pressure perturbation through optical coefficient and mechanical and optical thermodynamic and mechanical properties of tissue. So here we basically have a blend of optical, mechanical, and thermal parts, which also served somehow as a motivation for us to study in more detail these optical and mechanical properties of diatoms. Since within the framework of Russian Science Foundation grant, we are developing a bioreactor that would help us to automatically actually grow the diatoms under different conditions. We also decided to fabricate an optical setup which would help us to monitor their growth during this long-term cultivation. So in this case, we fabricated LED-based setup with a central wavelength of 505 nanometers, and decided to test the capabilities of monitoring the changes in diatom concentrations using 7 different diatom strains, centric ones, marine centric ones, and a freshwater pennate, which have different shapes, sizes, and volumes. And what we observe is the linear dependence between the light intensity and the concentration of the cell. In this case, we approximated the dependence at the linear fit and observed very good correspondence with this linear fit. But what is also interesting is that the slope in this case, in all these cases, are different. So that is why on this graph, you can see, for example, how we observed this monotonic dependence of the extinction and transmission slope on the volume of diatoms. And we believe that these results can actually help us in further tuning this optical system, to be able to distinguish between different diatoms, for example, if we have them in the culture. To sum up this first part of my thesis, so we performed monitoring of the growth of diatoms using fluorescence and photoacoustic visualization. The obtained signals exhibit a good correlation with their respective growth phases. Also, there are some advantages over these methods that we applied over traditional ones. First of all, it is rapid evaluation of the growth, which does not require extensive sample preparation. This is non-invasive, and it comes with a reduced risk of contamination. Of course, if we are talking just about the first initial growth phase, in that case, the preferable choice would be the standard cell counting method. We also performed monitoring of the changes in diatom concentration by fabricated LED-based setup and observed a linear dependence of measured transmission on the diatom concentration for 7 different diatom strains. This actually setup is portable, cost-effective alternative for diatom concentration measurements in bioreactors, but we also believe that it can be used, for example, in field settings and some resource-limited environments. Now, I would like to start with a second part, which is related to the study of mechanical properties and vibrational characteristics of diatoms. So we decided, as I have already said, to conduct this comprehensive study, which would include cleaned frustule without organic materials, with separate contribution from external and internal side of diatoms, as well as the diatom cell, which contains organic materials. I also mentioned the possible application in MEMS devices. Why? Because this whole idea actually originates from this striking similarity between the diatom valve and the microphone, MEMS microphones. For study of mechanical properties, we applied 2 atomic force microscopy approaches. First one is based on the acquisition of force distance curves, and second one is amplitude modulation, frequency modulation, viscoelastic mapping, which works in tapping, not in a contact mode. Also, we performed nanoindentation inside the column of scanning electron microscope to be able to observe the structural deformation of diatoms directly under the microscope, under static, but also under dynamic loading, which is also crucial for MEMS applications. And what is quite interesting is if we are looking at the nature, for example, we can find this frequent situation when a sharp indenter, for example, from this copepod comes into the contact with diatom cells. So basically, this situation presented here mirrors the situation that actually is frequently occurring in nature. For the purpose of measuring mechanical properties, we used the centric diatom, Cosinadiscus oculus iridis. The structure, the morphology and topography were studied with scanning electron microscopy and atomic force microscopy. So what we see here? We see here the diatom frustule, which consists of 2 overlapping valves, epitheca and hypotheca, that are joined together by these silica girdle bands. what is interesting here is the fact that it is a multi-layered structure with interlinked silica layers that differ in thicknesses, in pore sizes, and pore distributions. When we are looking at this topography, we can see that this silica frustule surface is not flat, but it's rather decorated with such porous domes and form a hilly terrain in order to better analyze and for better interpretation of the future mechanical experiments. And let's start with the atomic force microscopy results, which were conducted by maps. Here you can see just topography, and then here Young modulus maps of cleaned inner surface of diatom, clean outer surface, and wet diatom cell. Our results actually showed that Young's modulus in these thicker parts, not in the pores, in the case of cleaned inner surface is around 15 gigapascals. For cleaned outer surface is lower, it ranges from 10 plus minus 4 gigapascals, and in the case of wet diatom cell, we have a higher mechanical response, around 25 gigapascals. So why do we observe such differences in mechanical properties? The logical explanation here is because of the organic support in the case of wet diatom cell. So we have the organic materials trapped inside the silica frustules, which can affect their mechanical properties. But why do we observe differences here between the cleaned inner and outer surfaces? First of all, it is because of the thickness. So this layer, inner layer, is thicker than this outer surface layer. Also, it turns out that this layer serves as a foundation for building other porous layers during the last stages of frustule morphogenesis. But because of this very thin outer porous layer, cribrum layer, we decided that it would be more appropriate for us to not only evaluate these mechanical properties based on standard force distance curves, but also to apply a more gentle, this viscoelastic method here. And what do we see? Again, comparison between cleaned frustules and diatom cell with organic components. We observed that these Young's modulus values actually profile along these white lines shows values that are up to 2 times higher than values obtained using the standard force distance curves. And it may be explained by the fact that the force applied during these force distance curves in the previous case is in the micronewtons range, while in this case, it is in nanonewtons range. We didn't quantify it, but it is nanonewtons range is actually based on the literature data. So it means that we have actually different volumes that are deformed in the case of, for example, force distance curves where the volume is a lot larger. It may also include some void or void parts which will influence the Young's modulus values. While in this case, we have volume that is like few cubic nanometers. It is only in the surface vicinity. So basically we can say that it does not include any void parts. That is why we obtained, in our opinion, why we obtained these higher Young modulus values. So let's now see what happens if we deform the complete frustule under the scanning microscope using nanoindenter. I'm not sure how to video. This is a video. Can you help me, please? Yes. Thank you very much. So this is the result of dynamic loading. So first we see how the indenter approaches the diatom frustule. Then you see that after approximately 300 seconds, the frustule starts to break along the one edge. And then we apply the cyclic loading, and we see how this frustule actually oscillates during time without complete rupturing. So although in the initial we have some this rupturing along one edge, yet it stays, continues to oscillate without complete rupturing. And based on such behavior and results, we calculated the compliance, which is the inverse of stiffness, as a function of cyclone number. So we see here that the compliance increases with the increasing cycling number, and the average values were 0.019 meters per Newton. Actually, these values, according to the literature review, are in the very good range for example, MEMS microphones, especially graphene-based MEMS microphones. The next thing is the static loading, where we compared actually the response from dried cells inside the column of scanning electron microscope and cleaned frustule. In this case, we can say that these first four diatom dried cells actually have the same sizes. On the other hand, I'm sorry, you can't see here, these sizes were slightly different. And in this case, the indenter actually approaches from the girdle point band point of view. So when we compared the values of the forces on in the case of dried cells and cleaned frustules, we observed a lot higher, of course, values from the dried cells, considering this organical support from within. But why do we observe differences if we compare the cleaned frustules? One possible explanation may be actually explained by this figure here. So when we cut through this frustule structure using focused ion beam, we observed a very interesting situation. Actually, you can see here, like there are different diatom frustules trapped inside one diatom frustule. This situation is not regular. It can mean that there are some abnormalities that occurred during this whole morphogenesis process, but still it requires further investigation. The next thing we did is to perform vibrational characteristics. Why? Because this whole relationship between structure and mechanical properties influences the vibrational characteristics of the diatom frustule as a complex micrometer-sized mechanical system. So vibrational actually resonance frequencies of diatoms were subject of research before, but only in terms of numerical simulations. We decided to actually not only perform numerical simulation, but try to experimentally validate those findings. So what we did here is to use again the same model as previously, Cosinodiscus oculus iridis, to perform COMSOL simulations of resonant eigenfrequencies, taking into account its geometry, density, Young's modulus, and Poisson's ratio. And then we applied atomic force microscopy to be able to experimentally prove or check these resonance frequencies. For that purpose, we prepared the sample as shown here by welding the diatom frustules with different here diameters on top of the AFM silicon cantilever chip. And then this whole procedure was followed with sputtering with a thick layer of gold or tin, 30 nanometers, to increase the laser reflectance. So we applied two different methods based on the collection of frequencies of thermal vibrations, as well as by external modulation by exciting the piezo driver. Also, I would like to say that yesterday our article got accepted for publication in Applied Physics Letters. So finally, today I changed the status from under review to accepted. So what we see here actually the spectra that demonstrates experimentally obtained eigenfrequencies of diatoms, depending on different positions of this laser spot on diatoms. So these color bars actually show at which frequencies we obtained the values exactly as numerically predicted. And these numerical calculations were shown here and corresponding vibrational eigenmodes. We also calculated the quality factors, which range from around 21 to 90, which is of course not large enough if we are thinking about high sensitivity. But for example, in some applications where we preferred not high sensitivity, but broader bandwidth, these quality factors may be useful. So let me sum up this part of the thesis. We performed mechanical examination of Cossinodiscus oculus iridis, wet cells, dried cells, as well as clean frustules. We obtained Young's modulus values of wet diatom cells around 25 gigapascals, which is a lot larger than for the clean frustule, considering the support from the organic material. We also obtained about 2 times higher values of Young modulus collected by this amplitude modulation frequency modulation AFM method, which is due to the different applied forces. We performed for the first time dynamic cyclic loading of diatom frustule under the indenture in scanning electron microscope, which is indeed important for MEMS applications. Calculated compliance in the range of 0.019 meters per Newton is comparable to graphene-based MEMS microphones. We also calculated numerically resonance frequencies of diatoms, which are in the range from 1 to 8 megahertz, and these results were confirmed for the first time using atomic force microscopy. Quality factors in the range from 20 to 90 offer some versatility for applications that prioritize bandwidth over sensitivity. Let me now move to the optical properties of diatoms. So we are still at this single cell level. Before showing you some results of numerical calculations, I wanted to provide some context to better understand what happens when light propagates in water. So diatoms are phototrophic organisms, which means they need light for their proper growth and well-being. They live in aquatic environments at different depths. That is why we see here a solar spectrum at different depths. We see that at the surface level and depths up to 1 meter, usually visible spectrum dominates, where these red and yellow wavelengths are absorbed quickly than blue and green ones. But as we go deeper into the water, basically we have only this blue light, as also illustrated here. Diatoms live in the so-called euphotic zone, where there is enough light. It depends on water clarity, of course, and different dissolved mineral components, but it is usually up to the several tens of meters. This diatom that we used here is marine species collected at the Sea of Japan, and it is planktonic species, which means that it soars in the bottom column, but at depths up to, let's say, 50 meters. They were collected actually at a much less depth, around 1 to 3 meters. So for our future calculations, we decided to use the wavelength 420 nanometers. Another explanation for this choice of wavelength is because of the extinction spectrum of chlorophyll A, which is the main chromophore in diatoms. So we see this absorbance band here, and actually besides chlorophyll A, diatoms also contain another chromophore such as carotenoids. The main one is fucoxanthin in this green region of spectrum, also chlorophyll C. But if we are looking at the spectrum obtained from the single frustule without organic components, we can see that there is practically no peaks, again, which is of course expected because there are no any pigments as illustrated here. So for our study of optical properties, we applied a Fourier model method This numerical model of Coscinodiscus oculus iridis was actually created based on the previously obtained scanning electron microscopy images, as well as AFM topography images that I have previously shown in the where we studied mechanical properties. So basically, this numerical model shows us these all layers that that are actually here presented, and the different dielectric permittivities. So first, I wanted to show what happens with reflection and transmission in the case of diatoms. So here, of course, the diatom frustule in water is subjected to the incident plane waves, which means that we can use the expansion into this plane of these plane waves into Fourier harmonics. Depending on the number of Fourier harmonics, we will obtain better quality results, better images. So in this case, all our results are based on truncation to 97 Fourier harmonics. It is also to achieve like balance between computational efficiency and still high enough resolution to be able to distinguish between all these elements. So we see here the reflection and transmission in all channels, in main channel, in diffraction channels. So basically here we can see that there are some diffraction peaks that actually originate from the diffraction. If we are looking at the main channel, we can see the signal, which is basically only at the incident angles close to the normal angle, so around 90 degrees. If we are looking at the transmission, and yeah, I forgot to say that in this case, it was quite expected if we are looking at the differences between refractive indices between silica material, which is around 1.45, and water, 1.33. So very small difference between these refractive indexes actually explains why do we observe such image here. Also, the transmission, if we are looking at the region of wavelengths 400 to 500 nanometers, we can see the signal. Unfortunately, we cannot see the signal at higher wavelengths. And this is all presented on this transmission spectra, which in the dependence of the wavelength. So here we compared the such periodic structure of diatom frustule with a non-periodic one. But non-periodic one in this case means that it is a structure which does not contain pores. It has the same thickness and it is composed of the same silica material. And what we observe is that such periodic structure due to diffraction increases the transmittance of light in this region of wavelengths 400 to 500 nanometers by up to 2 times compared to a homogeneous layer of the same material with the same thickness. Why this region of wavelengths is important to us? Because, as I have previously shown on some of the slides, the absorption spectrum of chlorophyll A, actually, the chlorophyll A has absorption minimum in the, absorption maximum in this region, while water has the absorption minimum. So it looks like that from the optical point of view, it is quite good for diatom to have such perforated structure to be able to better utilize light. And then we performed numerical simulation of electric energy distribution inside the diatom frustule by using this incident wavelength 420 nanometers. In this case, on the left, you can see what happens when our incident angle is 90 degrees. While in this case, the situation is different because we have another incident angle. So basically, we observe here interference pattern which actually comes from this diffraction by periodic structure and symmetric structure of diatom. This pattern looks similar to the so-called Talbot interference pattern, which represents these replicated bright spots and dark spots under the frustule. So yeah, here we are just showing separately vertical cross sections, and here are just the horizontal cross sections and the specific zeta coordinate. Because we observed such differences when, depending on the incident angle, and because we know that in real aquatic environments, depending on the orientation, this the angle can be very different. We decided to integrate it to integrate actually electric energy distribution over all incident angle and overall polarization. And we here considered two separate structures. The first one, structure one, is structure with cavity. With cavity, it means that we are actually having frustule and we are having this distance here is 25 microns between this upper valve half and lower bottom half. While in structure 2, we don't have any cavity. It's basically like a structure with one valve. So what do we see here? We see here a maximal electric energy distribution depending on the zeta coordinate. And we compared these values for both structures with cavity and without cavity. In the case of structure with cavity, if we are comparing not only maximal, but also mean electric energy density distribution, we see higher intensity. So basically the maximum peak can be observed at this specific coordinate 3.8 microns, and we see that the values for this structure with cavity is 1.7. So it means that it is 1.7 times larger than incident wave propagating in the water. In this case, we obtained also values higher than 1, around 1.2 for the structure without cavity. And this all phenomena actually arises from the reflectance and the Fabry-Perot resonances that arise. In the case of a mean electric energy distribution, we obtained the maximum at the coordinate 2.3 microns. The enhancement is 1.30 times fold, while for the structure without cavity, we observed values less than 1. So actually this exponential decay of electric energy distribution can suggest that such structure, when we don't have a cavity, is actually a very weak waveguide. And here you can see what happens if we just take a vertical cross sections of these distributions as well as horizontal cross sections at these specific zeta coordinates. So we again observe this interference pattern in all these cases. And why is that important? Special distance from the top of the frustule. One explanation, or maybe not explanation, but interesting time imaging microscopy images of one single diatom cell, we can see the chloroplast distribution in ideal situation located just under the cells where we observed those bright points. Of course, there are several issues or challenges here, because it turns out that depending on the light intensity variations, these chloroplasts can migrate inside the cell. So this is like ideal situation, everything is perfect. But if we are having high intensity illumination, that these chloroplasts tend to aggregate, accumulate over the nucleus in order to protect them. However, all these results require further investigation to be able to properly say that indeed such periodic structure influences the light utilization by chloroplasts inside the diatoms. I would just like to sum up this part. So we applied a Fourier model method to numerically study the optical properties of Coscinadiscus oculus iridis in water. Such periodic structure enhances the transmittance of light in the region of wavelengths 400 to 500 nanometers up to 2 times compared with a non-periodic structure with the same material and the same thickness. And this is the place where we have the optimal chlorophyll A absorption for photosynthesis. The simulation results reveal these distinct interference patterns of electric energy density that indicate efficient light concentration within the frustule and potentially enhanced utilization of chloroplasts. This actually the analysis shows us that there is the impact of such cavity-containing structures that enhance both maximum and mean electric energy densities, while structures without a cavity exhibit rather weaker waveguide characteristics. So this whole phenomena holds potential for energy enhancement applications. But of course, they require further investigation, not only by numerical simulation, but also we need to perform actually experimental studies to be able to confidently say that, yeah, indeed, such structure so strongly influences the light utilization of by chloroplasts and therefore influences their photosynthesis rate. And now we are moving to the last part of my talk. It's the modification of diatomite. So for the purpose of modification of diatomite, we used gold nanoparticles and applied combination of layer by layer assembly and freezing induced loading. So layer by layer is based on the electrostatic interactions between positively charged polyelectrolyte, polyallyl amine hydrochloride in this case, and negatively charged nanoparticles. We applied such deposition process up to 5 times. So we obtain 1, 2, 3, 4, 5 PAH gold nanoparticles by layers. And then we used scanning electron microscopy to actually see if there is any difference in between those bilayers. So when we are looking at just one bilayer, we see the insufficient coverage with these gold nanoparticles. But if we increase the numbers of bilayers, therefore we can also observe that these gold nanoparticles tend to accumulate and tend to distribute themselves on the pores of diatomite. Here you can also see the results obtained by performing scanning transmission electron microscopy and transmission electron microscopy on the sample, which consisted of 3 bilayers. So again, we see very good coverage of diatomite by gold nanoparticles. These peaks from EDX spectrum originate, of course, from oxygen, silicon, gold, and copper is from the TEM grid, and maybe some other peaks can be due to the residual organics from diatomite powder. Since gold nanoparticles absorb well green light, 532 nanometers, which is the wavelength that we have in our Arsom Explorer 350, we decided to use photoacoustic imaging as a way to show the degree of coverage of these of diatomite by gold nanoparticles. So on these images, you can actually see the dilutions, the dependence of mean pixel intensities on dilutions for all samples prepared. What we can see is actually that the photoacoustic signal decreases with decreasing concentration of gold diatomite suspension, which we mixed with an agarose gel. And then we also compared the values, the maximum values obtained with the bare gold nanoparticles and extinction at 532 nanometers. So actually we see a very good correspondence, especially if we are comparing the extinction at 532 with the mean pixel intensity is in this green region. Maybe some deviations in the case of frequency range from 11 to 33 megahertz may be explained by the insufficient mixing and these accumulations of these particles in the center of these gel droplets. This whole idea with preparation of these gold-coated atoms was for the purpose to testing it as a SARS platform. So as a probe molecule, we used the rhodamine 6G and obtained spectra here shown, which actually demonstrates not only the spectra from gold-coated diatomite, but also bare diatomite and the rhodamine 6G itself. We see that there is indeed enhancement of the signal from rhodamine 6G, especially if we are looking at this 3 bilayered sample and 5 bilayered sample. We also calculated the enhancement factor and obtained the values around 6,000. So basically, we indeed showed that there is improvement of the signal, that such platform can be used, for example, for SERS applications. However, there are some disadvantages. So before I come to the disadvantages, the summary just on this whole part is that, yeah, the combination of layer by layer and phasing-induced loading method optimizes such spatial distribution of gold nanoparticles on the silica substrate. It actually creates numerous hotspots for enhanced SERS. The enhancement factor achieved was calculated to be 6,000. Photoacoustic imaging confirms the high degree of coverage of the diatomite surface by gold nanoparticles in samples with 3 and 5 bilayers. The advantages of using, potentially using this platform as a SERS substrate. First of all, the diatomite is highly abundant. Then cost effectiveness, eco-friendliness. Diatomite has high surface area. It is biocompatible. It is mechanically stable. Also, when it comes to the gold nanoparticles, we can tailor their properties, which can also influence the reproducibility of SERS enhancement. However, the disadvantages include reproducibility in nanoparticle coverage. So we did not achieve a completely uniform distribution of such gold nanoparticles on the surface of diatomite. Diatomite is a sedimentary rock. It is composed of different parts with having different sizes, different shapes. Therefore, the variation due to its natural composition can also significantly influence the obtained signal enhancement. And there is, of course, always need to assess the stability under, for example, harsh conditions. Now, because I have already demonstrated all these conclusions, after each chapter, I would like to ask for permission to refrain from rereading these conclusions again, but rather to concentrate on the future outlook and what can be done based on these research findings. So if we are talking about monitoring of diatom algae growth, we will continue with the development of optical methods that can be useful not only for monitoring their growth, but also their activity in carbon dioxide sequestration, in bioreactors, potentially in natural environments. This will be mostly done under this Russian Science Foundation grant. When it comes to characterization of individual diatoms, here we would like to start by fabrication and testing of diatom-inspired MEMS devices, so using biomimetic approach. We would also like to perform experimental study of numerically predicted optical properties of diatoms, and also to manipulate and try to control the diatom chloroplasts using optical tweezers. Also, we would like to explore the new fabrication techniques and different techniques for characterization and applications of the diatomite-based composites. Now, I would just like to show you the lists of all papers that are thesis related. So basically, during these 4 years, I published 14 articles were published or accepted with my participation. 5 other publications and 2 book chapters were also published. When it comes to conferences, I delivered speech at 13 international conferences during these years. And now, before I conclude this presentation, I would like to extend my gratitude to all colleagues who have been part of this research journey. Because I consider this thesis as a collective effort made possible by the unwavering support of my supervisors, the invaluable contribution of all colleagues who were there to always help me to participate in measurements, to teach me. I would like to thank jury members because during the thesis revision process, their questions, remarks, comments helped me to significantly improve the text. So it's not only about refining and polishing, but also about significant improvement of some chapters. So now I can say that I'm proud of these 295 pages. that I've made possible to obtain. I would like to thank all colleagues from Biophotonics, from HSM Labs, Russian Science Foundation grant for financial support. I would like to thank my friends, my loved ones, for their unending encouragement. And that brings me to the end of this presentation. Thank you very much for your attention.
Okay. Thank you, Julijana, for impressive amount of work done and nice presentation. Let's move on, and we open question session and start from questions from jury members, then audience here and in Zoom. So I would ask for all jury members to, let's say, ask question related to presentation or questions related to the thesis text, and please confirm whether you satisfied or not on the answers and modification of the text done by Julijana. So let's start in order how it appeared in the file changes log file 2. Please, Professor Kotelevtsev Yuri.
Julijana, thank you very much for your presentation. I'm sitting second time, probably because I was on a month ago on the talk which preceded, and we discussed things a lot, biological part, and probably I will repeat maybe my question from the previous talk. What fascinates me as a biologist in this model is that you have perfectly organized spacious mechanical structure, which is the result of the activities of proteins and pumps. And although you already answered that, I probably would like to ask it again. Could you, in few words, explain your understanding how this porous structure was obtained? What are the key players and key pumps in the production of the structure?
Thank you very much for your question. Yeah, this morphogenesis and organization of silica into such highly organized structure has questions that actually remains completely unsolved. Why? Because what happens is that this silica is actually taken from water in the form of water-soluble silicic acid. And then it is transported into so-called silica deposition vesicles when this whole magic happens. But how exactly those silica nanoparticles tend to organize themselves in a highly ordered manner was still not observed directly. So there are some indirect observations that prove that indeed there is so-called phase separation process that happens inside silica deposition vesicles that facilitates the organization of such structure. However, there are different groups that are now trying based on the different approaches that are trying to explain what happens on this level. So, for example, our collaborators from Limnological Institute of Russian Academy of Sciences in Irkutsk are working in this area by, for example, inhibiting the cytoskeleton. Which I found really interesting. Why? Because this cytoskeleton involves microtubules and actin microfilaments that participate in the whole organization process. If we are inhibiting their function, it means that we can in some way also obtain the information on what happens when this frustule is formed. This can be done, for example, by staining these microtubules and actin microfilaments and performing some live imaging using live imaging microscopy so that we can see how they assemble, disassemble itself. What happens if, for example, when we inhibit their function, what happens with those microtubules chains. Our colleagues already obtained some interesting results. However, they said that it is necessary to conduct more statistical data. So we need statistics actually to be able to truly say, yes, indeed, this whole silica organization can be performed in that way. On the other hand, there are other ways how we can actually try to see what happens. For example, if we are considering some environmental factors, if we try to employ maybe genetic techniques, although I'm not a specialist in this field, but I found some papers and it is actually so ongoing research, completely ongoing research in this area with the aim to try to observe these silicon nanoparticles and be able to completely say, yes, that is the way, this is what drives them, this is the most important thing. As far as I understood, there is no complete answer to this.
Thank you, Julijana. It's a whole lecture. I think that my question will evoke such a long answer, but you're really very well educated in this field. Thank you very much.
So, Yuri, are you satisfied?
Yeah, I am satisfied with the corrections and with the presentation.
Okay, thank you. And next, Professor Losic, could you please ask your question and answer the question whether you're satisfied or not with the answers and revisions provided by Julijana?
Thank you very much. Firstly, I would like to thank you, jury panel, to invite me for this assessment for this thesis, and thanks, Julijana, for a very impressive presentation. This is really brilliant work. And before asking your question, I would like to make a few comments about quality of your thesis and also on your revision. So as I stated in my comments about your thesis, this is a really outstanding PhD project in all aspects, from a number of papers. You have 4 papers as first authors, many as contributing authors, many presentation, very nice work, well done. About revision, in my opinion, all comments are addressed very well, making your thesis improved in many ways. You improve clarity, define much better aims and objectives, improve many technical details, providing more explanation, improving conclusion, and so on. So I'm very satisfied. So regarding questions, I really have a lot of questions, especially on optical property, but I don't have much time. I will raise only 2 questions. First questions is about application your lab scale method on real condition. As you know, diatoms are a foundation of food chain in this planet, and most important living species in this planet, sinking CO2, providing oxygen, and they potentially have huge influence of climate change. So it's very important to measure their level in oceans. So regarding your methods, my question is, is this method can be enough selective to measure a level of diatoms in ocean, considering that you have many other soft algae and many other species. So that's my first question. And second question is, in your conclusion, you made many, listed many achievements, and I would like to ask you, what's most significant contribution from your work to the current scientific knowledge to diatoms field and published work, and if you can explain why.
Thank you very much for such nice words. I am truly honored because you agreed to be my jury member, because the whole, when I started this research with diatom algae, I was first introduced with your articles, actually. So I started the study by reading your articles. So I'm truly honored to receive such nice words from you. So regarding the first question, Fourier model method is applied here because diatoms have periodic porous structure. I'm not sure that we can apply such method for example, other algae. That is probably the main disadvantage of this method, as it implies only on periodic structures. So we cannot include in this case scattering, which and we know that they are scattering objects, so probably that would also impose some challenges in application of this method to, for example, study of algae, level of algae, considering also some other, for example, green algae, cyanobacteria, and so on. If I correctly understood your question, or maybe you can repeat it and try to clarify it more if I did not correctly answer it.
Yes, you say right, but my question was on selectivity, because there is existing techniques that people using in ocean to measure concentration of algae, but that include all algae, so not only diatoms. So my question is, can this method can be selectively detect only diatoms that can be specifically used as indicator for climate change?
So regarding the actual optical monitoring of diatoms using fluorescence and photoacoustic imaging, I think we will not be able to distinguish. Why? Because in this case, we have only one, in the case of photoacoustic imaging, we have only one laser, 532 nanometers. And we know that all algae actually absorb well such, such wavelength green light. If we are talking about fluorescence visualization with iris, in that case, we choose specific filters for excitation and emission wavelengths. But again, the green algae, the cyanobacteria also have these fluorescence peaks at 680 nanometers. So I think that in that way, and considering also the resolution challenges of these microscopes, we would not be able. On the other hand, as I showed this fabricated LED-based setup, when we specifically decided to compare the transmission intensities in the case of different diverse species. So we looked at freshwater pennant, marine-centric diatoms that have different sizes, shapes, and volumes. So I think that maybe in that method lays the possibility actually to be able to distinguish, but just based on these so-called geometrical parameters. If we are talking about spectroscopic methods, again, there are peaks. For example, diatom algae has chlorophyll C, which is not present in the case of green algae. They have chlorophyll B. So maybe in that way, based on spectral measurements, we could be able to somehow distinguish between different species. Yet, I think that the selectivity indeed can be very challenging in this matter, to be able to say confidently that yes, this is indeed signal from diatom, which has such shape, such sizes, such volume, and this is the shape from, for example, smaller green algae, chlorella, and so on.
Okay, thank you.
And regarding the second question, the biggest achievement, well, I'm very proud of my very first article published in Photoacoustics, because we again proved for the first time that the photoacoustic signal from diatoms can be achieved. I'm also very proud on this dynamic loading by performing in situ SEM nanoindentation. Because we showed actually what happens when we subject the diatom frustule to cyclic loading under the indenter. I think I'm not able to choose just one accomplishment, because I'm looking at it like accomplishments from all my colleagues who helped me achieve these results, who just helped me to start cultivation of diatoms, because that is also a big achievement for me. I started this research 4 years ago, and I didn't even know what diatoms are. And now I can say that we are in our lab cultivating more than 15 different strains successfully. So that is also like big achievement, being able to work with living objects that all require some different conditions for their growth. For example, these bical species, they need low temperature. The centric species from Sea of Japan, they need like other conditions. So that is also somehow achievement, big achievement for me.
Thank you very much. You've done excellent work.
Thank you.
Okay. Thank you, Professor. Thank you, Julijana, for answers. By the way, to continue the question regarding selectivity, is it possible to use that eigen acoustic modes, like what you published in recent applied physics letters? Because it's should be too specific for
indeed. Yes.
diatoms.
A colleague from our group published the article related to the vibrational eigenfrequencies of diatoms 2 years ago, when he actually compared values from 2 different species. And they indeed differ because they have different geometry, different Young modulus, and it all influences these vibrational characteristics. That is why I think that based on these eigenmodes, we can distinguish between different diatom species. In this case, we only used one, which like centric diatom, due to its unique structure, abundance, because it can be easily cultivated. But we would like to try to use this model for example, these pennate elongated ones, to prove that indeed we would be able to distinguish between different species.
Okay, thank you. Let's move on. And next jury member, Dr. Ilya Turchin, please, your turn to confirm or ask more questions.
Okay, thank you, Julijana, for the very nice talk, and I got a great enjoy to read your PhD thesis. It was really enjoyed for me, and I am almost satisfied with their answers on my questions. However, I would like to ask a little bit more, and also that question about their absorption and extinction coefficient. So you've put in the initial PhD thesis version, it was absorption, then you change it to extinction. But however, may I ask to show the image of spectroscopic data of transillumination?
It's not working. I'm sorry.
Is it possible?
Elena, can you help us, please? Because it's not working.
Sorry.
Okay.
It's a little bit back.
Okay.
Okay, there are two previous, I think it's, oh, yeah.
This?
No, next
The
one.
next
Yeah.
slide. Okay.
So yeah, it's here. Thank you. So the first graph shows the extinction of living cells, and the second one shows the extinction of plain crystals.
Yes.
But so, do you think what effects dominate in absorption or scattering? Because the attenuation of light contributes from absorption and scattering as well. And what do you think on the first image and on the first graph and on the second graph? What effects are most dominated? The absorption or scattering, or it's impossible to discriminate between them?
Well, that's a very good question, because in the first case, we of course have this organic material. We have numerous chloroplast grains that of course absorb well the light, but I'm sure that they also scatter light well. In the second case, on the other hand, we have this porous structure, silica structure, which I'm sure that can not absorb well light like scatter. So maybe in the first case, the absorption part would be bigger, while in the second case, the scattering, just based on these like influences of organic materials and pigments inside the diatoms and the silica structure.
Yeah, that is what I expected to hear from you. And maybe you can just take, so you can say that the scattering is just defined by these frustals.
Yes.
And the absorption is defined by the living matter, yes, by the chloroplasts, because of the differences in absorption and scattering of both matters. Yeah. And the second graph is very correlated with the next slide showing transmissions.
This
one? Yeah.
Yes.
Yeah.
Yes, if we use logarithmic and inverse, yeah, we can obtain transmission based on that. But again, I wasn't sure that I'm able to do this because we are working, it is not completely absorbance.
It's just scattering. On that graph of transmission of clean frustals, you show their transmission through these frustals, and here is transmission again. So these two graphs are very well correlated. So you have the decay.
Actually, experimentally validated the numerical calculation.
Yeah.
Thank you.
Okay, thank you. May I ask some more questions?
Yeah, sure.
Oh, okay. So you have in the film measurements you have used to excite 402 nanometer and 660 nanometer. Yes, that's correct.
Actually, in this case, only 470 nanometers. Why? Because this was done on the other setup, not on the Peacock one that we have in Professor Drachov's lab. In my thesis, yes, I indicated the excitation wavelengths. They are indeed 402 and 638. In this case, this was done in Moscow State University Department of Biophysics, and they have a slightly different setup scheme. In this case, yeah, it is 478.
No, I'm appeal to that experiments you showed in thesis. You didn't show it in this.
Yeah, I didn't because I consider it like more descriptive, and due to the limited amount of time, I decided that maybe it would be better just to show this and not that one.
But in any case, did you find any differences in the lifetime?
Actually, no. I didn't present it in the thesis, but I have a lot of data on film, and we couldn't observe differences in fluorescence lifetime values after using 638 and 402.
Okay. Okay, thank you very much. I am fully satisfied with your answers and with your thesis Thank you very much.
Thank you, Dr. Turchin. And let's move on. Please, Dr. Cvjetinovic.
Right. Thank you very much, Julijana, for your presentation. A very nice presentation. And as everybody said already, which is not really surprising, it's very impressive.
Thank you very much.
PhD thesis as well. I have a few questions and a couple of comments as well. So if I may, I'll start with questions. So some of them just my curiosity about the subject. So you actually, so slide 35, if you go there, you showed the penetration of light in water as a function of depth.
Mm-hmm.
Do you know why blue penetrates deeper than the rest of the wavelengths?
Higher intensity?
You think it's intensity? Well, if you look at the spectra, I mean, the intensity is not that different, right? So it's clearly, it's not to do with intensity, it's to do with something else. I don't know what it is to do with, so that's why I'm asking.
It's a very good question, so I have to think more about it.
Well, it's all right. If you don't know, it's okay. I mean, you obviously based your work on this data, so not necessarily have some insight into why it is, but I'm just curious why it is. That's all. So the next question is, well, it's actually more a comment, so let me do the question. In slide 42, if you go to slide 42, you mentioned there chloroplast migration.
Yes.
You said something about chloroplast migration. What's the mechanism for the migration of chloroplast?
Actually, there is still a mystery that goes behind that question. According to literature, actin filaments actually are responsible for this movement of chloroplasts. We did preliminary experiments in Moscow State University with the help of the group from Dr. Cvjetinovic, and we actually were able to manipulate chloroplasts using optical tweezers. So during that process, I was able to observe those actin filaments because of the high resolution microscope they had. So actually, what is quite interesting here is that there are some old papers that also showed the possibility to manipulate using optical tweezers chloroplasts in spinach leaves, for example. But they said that it is impossible to observe those actin filaments. That is why I was so surprised to see that actually it is quite possible. We were able to see them. I decided not to include that in this thesis because those results were obtained in September, so I didn't have enough time to prepare it all. But actually, yeah, they said that the currently explanation is that the actin microfilaments are responsible for these mechanisms. But we will see what will happen next, because we are planning to not only experimentally try to catch and manipulate and control chloroplasts using optical tweezers, but also to again try to numerically calculate that to see what actually maybe there is some other mechanism behind those migration of chloroplasts.
Okay, so yeah, well, from what I understand, it's somewhat different, but I'm not obviously in the area. I don't know why was what was the need to do the optical tweezers. But to me, it's clear if they're migrating, there's clearly some feedback mechanism. So that would be something interesting to study. If you said it hasn't been studied. So I have a couple of comments also. So if you go to slide 40. Yeah, this one here. So you show this maximum, is it energy or transmission in this case? Energy density. Yes, it's maximum energy density is a function of direction. Just by looking at this, I don't know, has this been published, this result?
Not yet.
No, okay.
No.
So just by looking at this, I think perhaps Fourier transform will be very helpful because you'll have the frequency content, which will be, I think will be much more informative in design
then
of
this
waveguide and characterization of waveguides rather than just dependence of the energy in that direction.
Okay, thank you very much.
So the other comment is, where's another comment? It's on, yeah, so my last comment actually is on this fascinating question that Professor Cvjetinovic, yes, it's always difficult to get even your own name in Latin, was asking. And that's what is a mechanism? What is the driving force behind these intricate shapes? And you said that it's unknown right now.
Yeah, it's not completely known up to date.
Not completely known.
Because this phase separation process, according to literature, happens in these silica deposition vesicles. But without being able to observe actually those silica nanoparticles and how they arrange themselves in a highly ordered manner, all of these just look like some speculations, and without actually evidence, it's hard to say. Because unfortunately, this was not scope of my research. So just out of curiosity, I read some papers, maybe not enough to get the complete picture of the whole mechanism behind, because a lot of like these proteins, microtubules, microfilaments play a role, but we still don't know why and in which content. So yeah, unfortunately, I'm not sure what to answer,
so no,
because
that's what I said.
That's unknown, and maybe there are some difficulties how to understand, how to interpret the data, even with these inhibitions, because all diatoms are different. So up to date, there are around 100,000 species, and they all differ in sizes, in pore distributions, and so on. So for me, it is mystery how it can't be solved, because they are on this planet around 400 million years, 200 million years, I'm sorry. But we still don't know how they make those glass houses.
Yeah, as I said, it wasn't a question, it was a comment. And so my comment is, it's actually not surprising and not fascinating, because there are 5 billion people, we're all different. So to me, it suggests, from what I saw so far, that it's perhaps a combination of genetic factors, environmental factors.
Well, yes.
The next thing, they clearly live in communities, so they don't live alone. So probably there's a contribution there as well. So I would be looking at those 3 factors. I mean, I don't know whether the research into the origins of it all would be your future, but if you are, I would suggest looking in those directions. So that's all my questions and all my comments. And I can also state that I'm satisfied with the response to my comments to the thesis.
Okay, thank you, Dr. Sapelkin. And thank you, Julijana, for answers. Let's move on. Professor Fedotov, Vasily, please, your turn.
Well, first of all, I would like to applaud Julijana for a tremendous amount of work that she has done sort of in frame of this highly interdisciplinary research, which is always kind of a difficult endeavor. And also, I suspect that in the course of your research, there were kind of interruptions due to a pandemic situation, and so nevertheless, you have had so many results, interesting results. Also, now when it comes to thesis reviewing, I should like to think that I was particularly nasty to you with my comments, questions, and suggestions, and I would take my head off if I had one, you know, for a tremendous amount of work that you've done addressing all of that. Having said that, there are a few issues remaining, which I would like to discuss with you now. Well, one of my comments was related to chapter number 3. And so I suggested that it's probably better to move it to an appendix, but you decided to keep it in your thesis, and you kind of replied to my comment that this chapter serves as a comprehensive repository of various methods and techniques employed in the research. This is kind of fine by me. It's just probably would be better to include this kind of justification and motivation in this chapter in the form of a synopsis, because it's actually the only chapter in your thesis which doesn't have any kind of abstract or synopsis or introduction.
Okay, thank you.
Now, what else? Now, when it comes to chapter number 7, which deals with modeling light and energy distribution in the frustals, sadly, you haven't changed the title of the chapter, so it still reads electric field intensity distribution.
I noticed it late. Yes, I completely agree. Unfortunately, it was too late to change everything. Yeah, so for these mistakes is in the book. I apologize for that. I thought I'd changed everything into an electric energy. I noticed, yes, and I said, Professor Gorin, that I made a huge mistake, but too late now.
That's fine, that's fine. Then also in the text of this chapter, you seem to be using, so your terminology varies from intensity distribution, electric energy distribution, electric density distribution, light intensity, electric field intensity. So I'm just sort of invite you to kind of be consistent and work that out once and for all. And well, I guess you agreed with me that what you model is not the electric field intensity, which is E, right?
Yes.
But then if we're talking about electric energy distribution and light intensity, although they're related, I mean, they're both proportional to E modulus square, they're still kind of a little bit different because electric energy distribution, electric energy density is joules per meter cube, right? And intensity is watts per meter square. And I think when it comes to sort of discussing interference patterns and just distribution of light in photonic structures, it's kind of more common to talk about intensity of light, right, rather than electric energy distribution. Okay, what else? At the end of this chapter, you place a section which describes how chloroplast migration in diatom frustals controls the light intensity that they receive and so on. It would be good to still have kind of a link to all your sort of modeling results to see kind of a connection, because it seems like this information is there to kind of justify what you have been doing with all that modeling, but it's not really clear because this is an interesting fact, but it kind of remains sort of detached from from the bulk of that chapter. And the last kind of issue is that it is related to chapter number 8. There is a problem with numbering of subsections, because subsection 8.2 is then followed by subsection 8.5. So you'll probably need to kind of attend to that.
Thank you.
Right. And if I may have a couple of questions regarding physics and your work on those frustals. First of all, I would like to ask a very naive question because, well, I'm not a biologist. So
me neither.
Okay, so let's see if we can sort of work out that together. Your results indicate that the cells, after cell division, they begin to die, and one of the reasons for inhibition of the cell growth and death is lack of essential nutrients, because the nutrients are provided only at the beginning of the experiment, right? So I just wonder, why do cells choose to divide until they have used up all the nutrients and as a result begin to die? Why can't they adjust their life cycle depending on the nutrients available. Why can't they just slow or stop dividing before nutrients run out, and so in that way kind of extend the sort of life cycle?
Okay, if I correctly understood your question, I found some data in the literature that actually show that, yeah, diatoms are capable to adapt themselves to some medium where there is a lack of nutrients. In our case, when I obtained such results, and I noticed that, yeah, it correlates well with what colleagues from Limnological Institute in Irkutsk or Karadak Station told me, because they provide me with the initial samples. They said, yeah, indeed, 45 days, then after that we observed the dying phase. In the case of fresh water, in the case of marine, it's again around a month. But I wanted to prove just that I can use these methods for long-term monitoring of diatoms and cultivation. So, of course, that after, for example, 1 month, we just add these nutrients again, and therefore the cycle goes on and on and on. But in this particular experiment, I just wanted to see when this dying phase comes. For example, where is the start of the dying phase? One explanation, yes, it is due to the lack of nutrients, but also there are another explanation. For example, contamination from bacteria, because in our lab, we don't have completely sterile conditions. We are not growing oxanic cultures. So bacteria contamination can also influence the growth of diatoms, because they are competing for nutrients, basically in that case, for nitrogen and phosphorus, not from silicon. Also, for example, these in old cultures, if we are cultivating them, for example, for 100 days, some metabolites metabolite products actually can also influence the cell death. But it requires, I think, further investigation and maybe trying to get more statistical data, because I used just 3 different diatom species. Maybe for comprehensive understanding, we will need more, for example, conditions, or to provide actually sterile conditions without influence of bacteria. So yeah, that's like a question that I think that there are still some challenges that needs to be solved in this particular matter, to be able to completely surely say, yes, that is because of that.
Okay, thank you. And I have a couple more kind of physics-related questions. So can you probably go back to slide 38? Yeah, so here you sort of came up with the conclusion that due to diffraction, the periodic structure of diatom fossils enhances the transmission of light in the region between 400 and 500 nanometers by approximately 1.5 to 2 times when compared to a homogeneous layer of the same silica material. So I would like to challenge this kind of explanation in the following way. So can this enhancement of transmission be explained just by the fact that when you drill holes in a slab, in a very thin slab, you can effectively reduce the effective refractive index of the slab because you remove the material, right? And so since the effective refractive index decreases, then the Fresnel coefficients, so the transmission becomes higher and the reflection becomes lower. So is it really a case of diffraction that helps to kind of channel more light through the structure?
Well, I must admit that I didn't think about it in that way. But now I see the logic, and potentially, yeah, it may be possible that not only due to diffraction, but also due to these changes in refractive index. I think that we should then take that into account as well, because I thought that the main effect is due to the diffraction only, but not due to the refractive index. Although it's
well, you have such a huge amount of data. I'm sure there will be some that kind of either support this hypothesis or
okay. I will try to use the old data to somehow elaborate more on this.
And the last question, you sort of mentioned that nanoparticles, so this is, I think, the last chapter, that nanoparticles, well, during the deposition are preferentially located at the edges of the rim. And I think that was also illustrated on slide 45. I just wonder why this is so. I mean, what kind of controls this this process? I mean, why they choose to kind of sit around the edge?
Well,
maybe this is not so kind of obvious after kind of several deposition, but during kind of first couple of deposition, I think you mentioned that in the thesis that they
well, actually, during the first deposition, I cannot say that they tend to accumulate just along the edges. But for example, if we are looking at here, we also have somehow just looks like an uniform coverage exactly in this place. But here, maybe it can be also explained by the fact that diatomite is a rough material, maybe due to somehow surface roughness that is also around these pores. So they tend to somehow find the optimal place along these edges, along pores. And then after each deposition cycle, they actually tend to accumulate just beside those, it's so-called the favorable places, actually, upon deposition of polyelectrolyte and nanoparticles. So if, for example, they accumulated along the edges during one deposition cycle, they will tend to, upon next cycle, accumulate just along these nanoparticles that were along the edges. That's somehow an explanation that I found in other articles that use layer by layer, not for that purpose, but for fabrication of microcapsules. This is something that we also are doing currently, as well as biophotonics lab. And that is basically how I got the idea that to maybe, while I was fabricating microcapsules, I got the idea to use the same method to modify the diatomite surface. It just comes out of my curiosity, actually, but it turns that indeed we can use such methods to modify the surface, and it also brings us the enhancement of the signal.
Okay, yes, then I think that's all for me. Yeah, thank you very much for the answers.
Thank you.
Chairman,
Thank
if
you,
I can make
Professor
comment?
Fedotov. Yeah,
I would like to make comment.
Yes, please.
Okay.
Who is talking?
I saw a lot of comments about.
Yes, please.
Can you hear me?
Yes.
Can you hear me?
Yes.
Okay.
Yes, we can.
Yes, please. Go ahead.
Okay, I saw a lot of comments about optical property. I worked on this problem very long time ago. So it's very important to consider here a plasmonic and a waveguiding property of structure of diatoms. So when I done experiment hitting only diatoms with a very small spot of laser 1 microns on edge of diatom structure, all diatoms are start to turn on, all chloroplasts. So it means that we have propagation of lights horizontally, and that is a reason why diatoms are so successful to taking lights from in water. So because they have other means how they enhancing absorption of light using photonic crystal property. And it's good if Julijana can explore that in future.
Okay. Thank you. Yes, thank you again, Professor Fedotov. I think when Julijana will publish a book based on this material, she will use your comments.
Yeah.
And I saw some comments. I will give you a chance to. No? Okay. And now it's my turn. Actually, I completely satisfied with many of my comments. The only concern still exists is about fluorescent lifetime imaging, microscopy, and why I stress out this point, because I saw many papers from biologists just to show a picture and then take average over image average lifetime. But why do you need microscopy if you just average everything? Your nice picture you showed over there it shows strong distribution in lifetimes. It means that you have certain interaction, certain like environment which may affect lifetime, and then in the analysis you should take into account that like if you want to make conclusion, you should measure not only lifetime but quantum yield, so that you can distinguish between radiative, non-radiative decay. And one of the strongest features of the film that you can analyze kinetics, whether it's like double exponential decay or single exponential decay. So that's very important, and you have a lot of material, and I'm satisfied with your answer that it's like it's a matter of future research.
Yes, indeed. I hope I will have access to pick a quant film.
Okay, and I agree with our colleagues that when you consider transmission and extinction, you should analyze not only absorption and scattering, but also specular reflection. I missed that spectra for reflection. You measure only transmission, and as Vasily mentioned, okay, you change something, reflection, specular reflection is changed. So that's also. And one minor question, just for curiosity, do you have any idea or estimate what is the synthesis time in nature for these objects? You are trying to reproduce, but how long it takes in nature to synthesize such kind of structure?
Oh, that's a very good question. I mean, I know about division. So from one single cell, when we obtain two daughter cells, but regarding the whole process, I'm not sure.
No, okay. Thank you for your
4
answers.
hours.
And now we are open for questions from audience. So please go ahead, and any comments, any questions? Do we have any participants? Yes, we have some 13 participants online. Any questions from Zoom?
I can see none at the moment, but maybe let me make another call.
Okay, so if not, let's move on and ask supervisors provide some words about work, about maybe you have a question.
I don't have. Thank you. Okay, dear colleagues, dear guests,
I would like to start from unusual way. From acknowledgement to the Professor Salimon, because 4 years ago, Professor Salimon invited me to Julijana and Julijana to me. And as results, we have in our lab brilliant scientists together with very interesting objects. And Julijana is playing a role as bridge for 4 years between our groups and groups headed by Professor Karsunsky. And I would like to say that Julijana was working very hard during 4 years. And as a result, she has received many very interesting results. And this results is enough for obtaining many of publication. And I would like to pay attention that to the communicative quality of this person, because Julijana has also 5 publications are not related directly to her PhD thesis. Therefore, it's not only communications, but also skills that she has received during PhD program at Skoltech. And next one, Julijana was working during 2 years as TA assistant in the frame of our 2 courses, Physics of Colloids and Interfaces and Biomedical Applications of Photonics. And agree Julijana's efforts, we drastically increase the quality of our courses, and Julijana always has received very good positive feedback from students and PhD students. And also, I would like pay attention on the very high level of responsibility of Julijana. Therefore, she is the main executor of our Russian Scientific Foundation project. And the topic of this project is directly connected with her PhD thesis. And I would like also share with you the last achievements of Julijana. It's accepted publication in APL, and also Julijana was selected for White Vault Congress. That this Congress will take place in Singapore in January next year. It's this very famous Congress for young students. And 30 Nobel Prize laureates will take part in this conference. And I hope Julijana will have ability to communicate with such type of professional scientists. And also, Julijana will continue her... yes, it's next statement. I hope that Julijana will obtain also Nobel Prize. She has everything for this step. And but in near future, it's for long-term task, but short-term task. I hope Julijana will occupy next week position research scientist here in Skoltech. And also we have already discussed with Professor Karsunsky that I hope in the near future Julijana will be assistant professor here in Skoltech, and I hope she lead the very nice and very interesting direction in the from point of view research and from point of view teaching is direction related to bionics. It's very promising direction, and I wish Julijana very good future in science. And also I would like to say many thanks to our jury member for your evaluation of this work, for many comments, many ideas. Thank you so much. Thank you for your attention. And floor, I think I go the mic.
Yeah, Professor Karsunsky.
Yes, to Professor Karsunsky. Would like to go to mic. Yeah.
Well done, Julijana. I'll start by saying that Julijana produced a remarkable piece of work, which, for simplicity, we can assess using the familiar Oxford triad. Oxford Doctorate of Philosophy is probably the oldest of them all, predates Cambridge and is written slightly differently. But you know, when you have a very old way of distillation, it reduces to a very simple formula. A piece of work presented for degree must be personal, original, and substantial, right? So personal is, we've checked that Julijana did most of this work, although she collaborated, and that's another ability that people acquire during PhD. Original, we know, again, and substantial, but I would like particularly to point out the interdisciplinarity of this. I mean, as correctly pointed out, we are not, Yuri has left, biologists. Consequently, when we talk about these systems, we sometimes use such naive formulation like, why doesn't it do this? Why doesn't it do that? As if it has conscience and mind of its own. In fact, of course, it doesn't. Even though, as Darwin said, beautiful quote, by the way, the process of selection pushes it in a certain direction. So I feel that it's been, perhaps Julijana is lucky because we've had good luck following our progress in this direction. It started with Alexei's excited recognition of the potential of diatom topic, which he brought home here. And then with Dmitri's involvement and Julijana's eager ability to work on this, we built this project. I think the point about the fact that it's as deep as a book is quite right. And in fact, I'd like to say that I've seen theses which are 1/4 of this successfully defended in Skoltech. I've seen theses which are 2/3 of this successfully defended in Oxford. This is a very substantial, significant step forward in understanding these systems in a multidisciplinary all-round way. And all I can say is upwards and onwards. There is still a lot to discover in this beautiful system. We still don't know all about it. Thank you.
Thank you, Professor.
Thank you.
Okay, anyone wants to add a word? No? Okay, in this case, according to procedure, we should have a discussion among jury members. And so it means that online participants should also go to breakout room. Dear participants, dear jury members, let us finish our procedure and congratulate Julijana. Sorry for long coffee break. It's not because we debate anything. It was a technical issues. Still, we cannot connect with Professor Losich.
He is with us.
He is with us. Okay. Great. Congratulations to the connection system as well. So, Julijana, I would express our common opinion that you have done excellent work, and you got strong interest from jury members reading that and providing some suggestions, comments, and some minor suggestions still there in the and you will have to fix it in terminology or something else. It will be transferred to you. Anyway, with those minor corrections, only improve the text, not content. We congratulate you, and we are glad that you have done a lot of work inside school tech. This is great, and congratulations.
Thank you.